Okay, so should I start?
Yeah.
Okay, so hello everyone. So as you heard before, now it's going to be a presentation about the spatial constraints or the spatial aspects of microbiome dynamics. So I'm going to introduce these spatial dimensions shortly. Then I will talk about agent-based modeling and cellular automata models. And then I will show you a couple of simulation tools or packages and softwares. So when we talk about spatial dimensions, of course, we may think of several scales. So we can think from large scale patterns of, how microbiomes are composed and how it affects their compositions, interactions, and dynamics, or like smaller scale as societies or households. And then you can come down to one single host, which has several compartments and they are somehow connected. So these little small communities are separated, but still connected via different nutrient flows. And then, of course, you have the very small scale dynamics. There's a question, but not to me. Okay. In the chat. So the small scale, which is really the cell-to-cell interaction between the different microbes. And of course, you can also think of having communities that are more well-mixed, so they don't have a concrete spatial structure, or they can be seen more like well-mixed communities, but then you have these spatially constrained communities with very discrete spatial structures. So you see here how the different models, so there are different models, right? So we heard about population level models like the Lotka-Volter models yesterday. So these were non-spatial models. Then you can have some sort of spatial models, but still equation-based model systems. And then if you want to really include very fine detailed spatial structures, then you go for the individual based models. So now I'm going to talk about a little bit about these different models that you can use. So these are mechanistic models. So they are based on these different dynamics and the mechanisms that we build into models and see what kind of collective behavior emerge simulating these models. These are different than these statistical models that try to capture different aspects of microbiome compositions. So when we want to include or want to consider spatial patterns, there are different tools, of course, not just individual based models, but you can think of metapopulation, meta-community models, or different network models. So it depends what resolution you want to capture. So in the case of host-associated microbiomes, you can have meta-community models. So these are not necessarily agent-based models, but you can have different compartments. These are the local communities which are connected via different dispersers or even, flows of nutrients or other things. And then you can also model heterogeneity of these compartments of these communities and then different dispersal rates. And then, as you see, the compartments can be filled up with microbes, but also you can model pools which are not the microbes within the hosts, but from the environment and how these exchange, different species. And of course, the most realistic model is when there's a host feedback. So the host can also affect its microbiome composition, which then in turn affects the environmental microbiome composition. So there is a feedback loop between these processes. The thing is that we don't really know how it works, or we are at the stage of understanding it better and better, how the small kind of ecological network with an individual is composed, how it builds up. So is it like individual-based dynamics or individual dynamics, or is it more like a group, so like a household or a small community shares some characteristics, or is it like universal? So you can consider these type of things. And of course, if you want to move away from this meta-population or meta-community models, then you can have some sort of networks. So you can consider microbiomes as networks within networks, or networks of different reactors, well-steered communities, and they are somehow connected by flow. So you can imagine this is more like a gut microbiota, or you can have some sort of spatial structure or ecological network within each community, and this is also connected like a linear network, and this is connected by flows again. But what we are going to discuss today in more detail is this microscale spatial patterns and aspects of microbiome dynamics. So this is, of course, mostly about, if you don't think about biofilms. So what is important here, that each cell occupies a different location, therefore it senses a different environment and is surrounded by a couple of other type of cells, so it has a different interaction environment. And to model this is not straightforward. So we saw yesterday how a model looks like. So you want to build up a simple model, then you assume that, for example, you have a prey and a predator population, right? So you have these dependent variables. You're interested how they change in time. And then you have some variables that characterize how these two populations interact, and then you build up these simple models, and then you have to solve them, as you saw yesterday if you were attending the lecture by István. So this is already not very easy, as you saw, but you can imagine that you have, if you add some more complexity and spatial structure, it becomes more complicated, right? So you can say, for example, prey can occupy different localities which have different resource, different amount of resources. So the growth rate might be different, or the predator has different, available prey within its neighborhood, and it can move only slowly, and the neighboring localities. So this was, of course, for another type of modeling framework, which we call individual or agent-based models. And I'm going to talk about some characteristic of agent-based models here. So maybe you have heard this, and I gave a kind of similar lecture last year, but I'm going to quickly run through the main features here. So agent-based models are, of course, computational experimental labs, we can say, in which you can create some environment, and then you fill up with agents which interact between themselves and with the environment, and then you can run experiments, numerical experiments with such a model. It is not a new thing. So the very first agent-based models came actually from in the 70s, as you can see, mostly simulating forest dynamics, and then it started to infiltrate into other areas of biology and ecology, and then into social and economic and engineering areas. But now it's basically used in every discipline almost. So we can, just a quick terminology, of course, we talk about agent and individual-based models. So according to my understanding, or how I see it in biology, maybe we talk about individuals, or in ecology, agent is a more general term. So agents can be banks and companies, but can be individuals. And sometimes talk about particle-based models, in which this subcellular level agents, basically. And then you can also see there are other terms that are often used, like multi-agent systems or cellular automaton models. So agent-based models are through 2- or 3-dimensional simulation spaces that has agents that are self-contained, autonomous. They have rules that they follow, upon which rules they decide how to act. So they want to reach a goal, and of course, agents, as I saw, can be heterogeneous, so differ in their states or rules or internal states. And then they have relationships, so they interact between themselves and with the environment. And of course, we have an environment which can consist of several things that the agents want to pick up or want to use, and they can also modify their environment oftentimes. So the agent is, as I said, is self-contained. They have autonomous system states. They perceive some clues or some signals from the environment, and then they decide on that. So basically, they are quite complex functions which have memory of past events. So this is somehow included or integrated into their states. And then they have some autonomy, some rules, how they decide what they sense in their internal states, what kind of actions they take. Yeah, so main features, it's autonomy, social ability, reactivity, and proactivity. Also, as I said, perception, performance, memory, and some rules, policies that they follow. So based on this, you define an agent, but you can also add more features like modularity, or you can have some sort of adaptational learning, evolutionary or social learning. You can have you can have scale separation, and you can add stochasticity and many other things to your agents. And based on this, you design an agent-based model. So first, of course, you specify the problem or the question what you want to answer. And then based on this, you define your agents and their attributes, their decision rules, what states they can have. And then you define the environment. What is it? How is it composed, and how agents interact with the environment and within the environment. And then you define what are the behaviors and the actions of the agents within this environment. How do agents interact with each other and with the environment? And finally, it's becoming more and more important. Then you try to validate your model, and there's some data that you can use to specify some of the parameters in your model. So you can basically, if you're building an agent-based model, you can aim for two things: abstract ABMs, which are basically very simple ABMs, but still they can capture very essential mechanisms or patterns. Or you can have empirical ABMs, which are much more complicated, much more complex. Probably you will have to have some data to validate them or to narrow down the probability of or possibility of parameter combinations here. So these are more complex but more realistic ABMs. And then if you start simulations, then you have to see that it's that the results are not so easy to analyze and decide whether what sort of outcomes you can expect and what sort of outcomes you can be satisfied with. So for example, it is always a question if there is some, so the dynamics arrives to a settle point. So is it equilibrium dynamics that you expect, or there are very long transients or non-equilibrium dynamics, and that is already an outcome. And of course, it depends oftentimes on the initial conditions, and there are many dependencies and multi-stable systems. And then, as you can see, many other things, but luckily, I mean, it's still cheaper to run experiments in silico laboratories than in real life. So we still want to use these ABM simulations to explore some of these parameter species and possible scenarios. Now, so we saw how an agent looks like. So now let's see how the environment can look like. So the environment can have some spatial structure, and these are we often call cellular automata or grid structure, but it can be also a continuous space, or you can also assume some sort of network topology or some map-based topology. But of course, you can also say that your agents are in a well-mixed environment, in a soup, in a water body, for example. So they are, we say these are the well-mixed models. And then when you have a cell-based modeling approach, then you can have the lattice models, of which the cellular automata is one type, in which one individual occupies one grid point in our lattice. But you can also define models in which you have basically a small population in the lattices. So these are these models where you have several individuals on a lattice, or you have these cellular pods models in which individuals are represented by this shape that can occupy several spatial points. And then, of course, you can have models that off-lattice models, and then these are models in which you track your individuals based on the center of each hypothetical cell, or some subcellular elements like different cellular organelles, or you can track them based on their boundaries. So, for example, the cell wall or their membranes, and then you calculate basically their shapes based on these tracking these different points. But what we are going to talk about is mostly the cellular automata models, which can also have many other shapes. So, for example, if you assume different topologies, so you can see here the cellular automata, the grid-like. So this is the spherical representation of the cells, or you can have the tessellation or the Voronoi type of representation of cells. So if you run the similar, for example, this is an aggregation experiment of aggregation and separation experiment, you can have very different results, assuming the same initial condition, depending on the topology or the cell shape you choose. So now coming back to cellular automata models, you have the lattice, you have the cells, which can, as I said, occupy one grid point. They can have the cells, or the lattice can have many states. So it can be empty or occupied, and then the cell, it can have many different states. And then you define the time. How is time defined? So is it based on cell movement or cellular processes, or is it based on, for example, diffusion properties of particles? And then the rules and the neighborhoods you also define. So the neighborhoods, or the dimensions of the APM, can have this rectangular, and this is the most common we use. So you can define the neighborhoods, like the von Neumann neighborhood, in which north, south, west, and east cells are the immediate neighbors. So this is with whom the central circle interact, or you can define a more neighborhood or even bigger surroundings around the central focus. But of course, it's not necessarily rectangular. You can use triangular, hexagonal grid shapes, and then you define this neighborhoods based on this. And the other important thing is how you define the boundary conditions. So you can have reflective or absorbing boundaries, which means that cells kind of get cancelled at the boundary, or they can be reflected, so they kind of see it as a wall and turn back their movements. But most commonly, what we use is this torus. So when we make a torus out of it, so basically there is infinite movement can be done on this grid. So the individuals or agents never bump into a wall. But in microbiome research, maybe this is more appropriate when you only have the tube, and then you can see this maybe as a gut, modeling of a gut. The update can be synchronous or asynchronous update, and depending on the question you ask, it can bring you very different results. So in synchronous update, you update every single cell at the same time step. In an asynchronous update, you make your updates random on the grid. And besides one layer of agents, then you can have several layers defining different type of interactions and different type of aspects of them, for example. And then you can also differentiate time scales. So as you saw, we have talked about already the diffusion. So the small molecule dynamics is much faster, diffusion of different chemical compounds. And then you have the mechanical interactions like cells bumping into each other and pushing each other. And then you have the slowest dynamics, which is the cell growth and then the division, for example. And then you can have also multiple different type of agents. So you can see multi-level cellular automata, for example, define them as the host and the symbiont, and their interaction is within between the host and the symbiont, and then they compete, of course, with their within their own habitat. Or you can have one host and multiple symbionts modeled in this spatial explicit agent-based models. And then you can, as I said before, you can have a combination of agent and particle-based models. So you can model individuals that produce some sort of compounds. In this case, it's antibiotics, and then these diffuse quickly, and then there's some sort of fighting between the individuals that produce and that are sensitive, but better competitors in sense of growth rate. So you can see one overtakes the other, depending on the diffusion properties of the system. And then you can also move one step towards reality, considering, for example, the gut microbiome. So you can have a gradient model, and then you can say, well, I have a gradient and I have a certain direction of flow, and then you can model flow of different nutrients, food through this colon, and the bacteria, and how bacterial density changes over time with these flows, and how it looks like at different time steps. As I said in the beginning, there's some challenge in validating ABMs, especially if they are complicated. So if you have a data set, of course, you can split the data set and train your model with one half of it, and then simulate and test it with the other half. But it's not often the case, but there are some papers that try to use this direction. For example, you can use different machine learning algorithms to try to define the interaction patterns in a community. So based on the colonies, their distance, which colony is near which colony and how they interact, you can say that these are more like mutualistic communities or cross-feeding types, or they are antagonistic, so they have negative interaction, and you can build up some sort of interaction network based on this. Or you can try to capture the colony growth characteristics. So if you have a system and then you have certain parameters, you can try to narrow down the parameter space based on observing real colony growth the characteristics, and then you can define your model based on these validated parameters. So summary of some of the models here. There are, of course, many more models. It's just one kind of table from a recent review. So you can see basically what I wanted to show here, that most of the models, of course, it makes sense if it spatially explicit. So you have a biofilm occupying a surface, but many of the models have metabolic interactions or physical interactions, but only few of them actually model the microbe-host interaction, which is an open niche. Of course, it's because these are more complicated models. I think we are developing these models nowadays. Now I going to show you some simulation tools and packages. How can you build up an agent-based model? So you can, of course, use specialized softwares that are specifically built to help to implement agent-based models in an easy way, or you can have a general purpose programming language like C or Pascal or anything, and then Python, and then you can use your models, define your models. But of course, you need some skills in coding to be able to code in this, or you can use combined solutions. And then, as I showed before, you can have the cellular automata models in which one cell occupies one location, or you can have the cellular pods model. We are not going to talk much about this, more like this cellular population or lattice gas cellular automata models, in which one small location represents a small subpopulation or so small subset of individuals from a whole biomass or biofilm. And the other distinctions that we can make is based on the methods. If it's rule-based methods in which we assume very simple rules, very simple decision behaviors and sensory behaviors and actions of cells. Or you can use equation-based models, so there is a complex metabolism and a continuum of behavior responses or phenotypes, as you can say. So one is simple classical building up maybe some sort of logic gates, how individuals can sense and react to certain inputs. The other one is what Agnes talked about, is more like some optimization algorithms calculating complex functions as biomass and metabolic flux balances within cells, and then you model your agents or your biomass based on these models. So these are some platforms that were already mentioned in the previous presentation, but I just wanted to show that there are some which have this well-mixed populations and some which include space in their simulations. And here you can also see another type of comparison. Here, what is interesting to see, for example, if evolution is modeled, maybe only in the Comets 2, the new Comets version, you can model evolution, but you can also see that these are the population models, and Bacterin and MatNet and IndiMesh are the individual base models. So I'm going to show a couple of these packages or softwares in the following. So the very simplest model is this rule-based bacterial modeling. And it's basically, as I said before, you have a grid with certain environments, so you can set the resource levels for each grid separately. And then you have artificial organisms or agents that sense the environment. So they have some detectors. So this is what signals they can sense. And then they have a rule which depends on the outside environment and their internal state, and they make decisions, and then it feeds back to the environment. It's very simple things, but you can make nice simulations. For example, in this simulation, You see that the lighter or whiter areas mean higher resources, and cells don't move here only by division, and you place a couple of initial founding colonies or cells here, and then what you can say that these are the white ones are the living cells, and the ones with the X's or crosses are the dead cells. So what you see here is that, of course, there's a lot of division in the resource-rich environment, but then once the resource is depleted here, all the cells die. So it's a very simple model, yet it gives us back some sort of heuristic understanding of colony and bacterial cell growth on a resource or a medium. And then there are more complex packages that you can use to set up spatially explicit models or cellular automata models. So, for example, this Repast is interesting because there are different versions of it depending if you want to use it on supercomputers, you want to run your simulations on supercomputers, or you want just to work on your desktop computer. Maybe a more popular one is this NetLogo you might heard of. So it's freely available. You can download it, or there's a web version of it, and then you can have different agent-based models. Of course, these are not microbiome-specific models. So these are the rule-based models. So you set up certain rules, and you can set up the agents that are called turtles here. So you see that these can be human agents, or fish, or virus, or fungi, or even fire. You have fire, how it spreads in a forest. And you can run this simulation quite nicely, quite nice visualization, easy to use. And of course, you can also use it for microbiome simulations. The next package is this just, which is interesting, or its special characteristics is what I showed you before, that it not only includes this type of cellular automata with the rectangular shape of habitats or grids, but you can also use the different overlapping circles, or the tessellation, or the Voronoi type of modeling for cells. So you can see here some of the modulation simulation results. So here you can simulate cell packaging in a tissue, for example. This one is the BASIM is basically based on, as you see also in this logical gates or simple rules. So based on the different resources, how it reacts, how it grows. And then you can simulate some colony growth based on these simple rules. But what is more interesting is that you can model physical cell shapes here, and you can see, for example, in this simulation that these physical constraints causes cell to push each other upon division or growth. So you can more realistically model, for example, biofilm growth or population expansion, in which movement is based on this physical pushing by the dividing cells. And then we are moving from rule-based to this equation-based models. So there is this MapNet, which is a combination of the NetLogo and the MATLAB packages, which contain this Cobra toolbox, this constraint-based modeling tools, and you can run simulations integrating these tool approaches in this MapNet. And then we have the Comets, which, as Agnes mentioned, one of the most popular packages used. So it's you can run simulations in a virtually test tube, batch culture, or on a petri dish in a spatially explicit model, and here one grid cell or one spatial location is actually a very small space which can occupied by a limited amount of biomass. So you don't have agents here. You have small biomass modeled in these grid cells, and then you can run this Comets based on their own user interface. You can run it from command line, or you can integrate it into Python or MATLAB. You build up your models, and then you do the calculations of the different optimizations for biomass or flux balances and stuff like that. That was already described by Agnes. So in the afternoon session, I will show a couple of examples how to use this on these different platforms and how such a model builds up. The next software is this Arena, which is based, again, it's based on spatially explicit models. In each cell, you have a small network, metabolic network, and then you can optimize model colony growth based on optimization for biomass, or you can model cross-feeding interactions. And you can, there's a paper that compared these two softwares, so you can compare them, how they perform given one type of syntrophic interaction that you see here. So these are the simulation results, and then basically this compares the advantages or disadvantages of one or the other. The next one, which I included in this presentation, is this three-dimensional dynamics flux balance analysis. It is interesting because it really can model colony growth in a three-dimensional structure like this. So this model looks like a three-dimensional lattice. So each lattice can be medium, like an agar plate. It can be occupied by cells, or it can contain air or oxygen. And then diffusion is like this. So in this 7 neighboring cells can affect the focal cell. And of course, you have passive and active uptake of different nutrients and substrates. And then you run the flux balance analysis of the metabolic network, which is interesting that you can switch on off certain parts of the metabolism. I will show you shortly. And then you have the growth or expansion of cells based on this, pushing upon splitting. And then you can have some, regulation. So, for example, in this paper, what you can see in these simulations is how a biofilm layers how different metabolic strategies are better fit for different parts of these biofilm layers. So this is an unregulated model in which cells do all the different parts of the metabolism altogether. And then you can run a regulated model in which, depending on what is mostly available for the individual cells at certain locations, you can run based on, so here you can see that the glucose is available because these cells are close to the agar medium on the bottom. So they run this glycolysis part of the metabolism, but they don't have oxygen available because they are not connected to the upper layers or the surface. So they cannot run the Krebs cycle, but for here you can see that some parts are switched on because they are close to oxygen, or these ones are the cells that can reach both necessary substances, both oxygen and the sugar, and then they run different metabolism, and then you can calculate how a biofilm can grow in a more realistic method. And finally, what I wanted to show is this IndyMesh, which was also mentioned previously. So here, another type of heterogeneous environment can be modeled. So it's mostly built for soil habitats. And then here you can also say that different type of compounds are available at different locations. So there's very complex heterogeneous microstructure here. And then the cells have access to different nutrients or different substances. So they switch on and off different parts of their metabolism, and then you can model them, and also their division is, so they can occupy these fractures in the soil, so you can model these paths in this modeling platform. But of course, there are many other platforms or packages or software. Some are for academic use, some are for commercial use or industrial use. Just a few are mentioned here. So this is all very nice and very rich and very can model many things. But question: is it the best tool, or is it the best thing that we can do? So there are, of course, some disadvantages of agent-based models. So of course, modeling large-scale systems is difficult. So in these previous models, you can model one small colony or 1 millimeter of soil or something like that. But really, modeling large-scale systems takes a lot of time and very demanding for computational resources. It's not an easy task. And of course, when they become complex, then you can just add one more module and they become more and more complex. It takes more time to submit. So it's all the simulation times and the complexity can be really high. And then you cannot always validate your data, and you don't know the sources of these different heterogeneities necessarily in these communities. So you have to make assumptions, but still parameterization and validation is really difficult. But maybe we have a hope that future technological and scientific and computational advancements will allow us to build more accurate and more realistic agent-based models. So you see here a couple of features which are, which would be good to have, like really modeling the signaling pathways, or having realistic evolution, cellular evolution, or dynamic cell shapes, which are not these Voronoi-based models, but really agent-based approaches. So with that, these are the references that I used. With that, I thank you for your attention. And if you have any questions, please shoot me.